you remember that for the tumors go from the first side to the second side to the circulatory system. In fruit flies, they don't have these beautiful highways. Yeah.
Okay.
Because they have an open circulatory system. So well, these ones. Okay. You can see my cursor, the mouse. So this one, imagine this like rivers, or streets that they can travel. In fruit flies, what you have is like the entire sea or a lake, where the pieces of tumors will be traveled all around.
Yeah.
So we don't know exactly what happened in flies. And we wanted to see if what we found is that the pieces of tumor they break, and we want to see if they are able to attach to another place. I don't know. Look, this is something that I will send you this presentation, but and we will talk later about this. I will send you my PhD thesis, and you can find hundreds of links about this. This is the fruit fly. Here is a pre-pupal stage, and this is the adult fly.
Yeah.
Do you see all these white regions of the pre-pupal stage?
Yeah.
All that tissue will die.
Oh, okay.
And these tiny sections will become the wings. These tiny brown sections will become the legs. So what happened there is that half of the larvae of the pupae die, and you have the very small regions that are called imaginal discs that they display a lot of proliferation, and it will be like a second morphogenesis. So I'm going to show you. I'm going to show you a movie of what is happening from these people. So look, these will be the legs.
Okay.
And here you have 1, 2, 3, the 3 pairs of legs. This will be the wings. Let's see it again. So this is what is happening,
Oh, okay.
Good. Yeah. And you can watch that movie hundreds of times. And here, this is the prepupal stage, and here is the pupal stage. This is in these two stages is happened this. Later, the movie that I have showed you is in between these two. The green was that I show you where the comb is rotating. I show you what happened in the leg. This is a more general stuff.
Okay.
So here, let me show you in a simple way. In blue are the legs.
Okay.
Good.
Yeah.
In red, here you can see this is a wing. But why you don't see the entire wing? Because we are imaging, we will have to turn the pupae in the side to image the wing. Yeah.
Okay.
We can only see this. Here there is a structure that it seems that it is separating this disc, the imaginal disc, the imaginal disc of the legs, of the wing, of this mouth part, of the cephalic complex that is separating them. So I call them distal boundary. And what we are doing here, I'm going to move you and move me here, is just we express, as we work with fruit flies, we are working with some genetic perturbations to produce tumors and see what happens. Yeah. So the green thing that you're seeing are tumors. Yeah, are pieces of tumors. And the question that we have is like why they are breaking and traveling all around, and what is happening while this happens, while they travel. And if you see, there is a lot of mechanical tension.
Yeah.
Because there should be a lot of pressure while they develop.
Yeah.
So we don't know exactly what is happening. So we want to see where do they travel to a specific region? Do they travel to in groups? Do they travel individually? What type of cells are all of them pieces of tumor? Maybe what are those cells? Do they move chaotically, or they move in a specific way? So the idea that you have to do is try to find ways to answer that question. Yeah.
Okay. Yeah.
So these are the genetic backgrounds that we use in RAS B12 and Scribble. This is not very important. These islet split paste, there are genetic techniques that allow us to make these tumors, and later we can talk about that. As I told you, there is so much things that slowly I will give you a piece of information. So look, will be the normal stage. What you will
Okay.
see is something like this. However, in our case, we only use a labeling for the tumors. For the legs, instead of using a GFP, we use a microscopy technique that allows us to see using shades
Okay.
to see more or less what is happening there. Yeah, to make it simpler to see if we see something. And in the beginning, you will not see anything.
Okay.
Slowly, you will be okay. There is a leg here. Oh, there is a wing here. Oh, they are going through. But don't worry. In the beginning, you will not see a lot.
Okay.
How they did that? Very easy. Very easy. Again. Oh, so generally, what I did is, you remember that I told you that there are some tissues called imaginal disc. So
Yeah.
there are a
Yeah.
group of 20, 30 cells that they start dividing like crazy. This is why
Okay.
these tissues are so good to study cancer, because they, in a short time, they divide like crazy and they display morphogenesis. What I did is I put the fly in a microscope and I was able to see it live. Yeah.
Okay.
However, generally, what people have been doing for years, to be honest with you, everybody told me that was not possible what I did. But the movie that you said, it was they said it's not possible to image those things, and it's possible to. But what people generally does is this they take the piece of tissue,
Okay.
they put it in a petri dish, and they put it in a media, and they make movies for 20 hours. Yeah, and then the
Okay.
tissue dies. Has
Yeah.
this have some advantage and have some disadvantage, because it's, maybe you don't know if what is happening there is because the tissue is dying, or because you pressure with your 4 steps, or what is happening. So I'm going to show you a movie.
Okay.
Do you remember? Look, this is a leg again, yeah?
Okay.
And it's going to elongate. Think about leg elongation, like uh, like when you have a telescope, yeah, that
Yeah.
you can extend like this. So this is like a telescope and it's going to extend.
Okay.
So let's see this and tell me what you see. Let's play this movie.
One thing I noticed is that there's this black region kind of thing that goes all over the leg,
Mm-hmm.
and it looks as if the leg is kind of like removing a skin or going out of something. Yeah.
Excellent, excellent. This is something very interesting. That is called a periporial membrane, and it's like, imagine that you have a
Okay.
sock, and you have to remove it.
Yeah. Yeah.
They have to do that. So if we made a tumor not only in the leg but in the periporial membrane, when they break it, the tumors
Okay.
will start traveling all over the way. And the question is like, what happens if you break these pieces of periporial membrane? The tumors are going to break and
Yeah.
they are going to die, or they can reinsert in other places. Yeah.
Okay. Yeah. Yeah.
Good. And
Yeah.
look, let's see it again, and you tell me what else do you see? Do you see dots traveling?
Yes. Like kind of like white circles.
Yeah, exactly. So this is not an artifact. We find it also in our movies. Yeah.
Okay. Yeah. Yeah.
So naturally, this happen. These pieces of tissue. Yeah.
Okay.
What generally happens is that it's going to other type of cells are going to phagocytize. They are going to eat these pieces of extra tissues,
Okay.
and however, if there are tumors, we don't know if they are going to be eaten or not. Yeah. And this is the question. This is some of the questions that we have. I already sent an email to this
Okay.
lady, that the author of this paper, to ask
Yeah.
her what is this. We don't know if these are immune cells. These are peripheral cells only. If there is a combination of cells, this could be pieces of immune cells too that are coming to eat and to destroy those cells.
Okay.
And they maybe if when you watch the movies hundreds of times and you say like, oh, and you will tell me, Nicholas, look, there is a, because this will happen in the beginning,
Okay.
I will tell you something, but after you watch it multiple times, you will see more things that I see in the movie. This always happen with all the students, and you will
Okay.
tell me there are different type of dots. Maybe this could be this cell. Maybe this could be this. We can make this hypothesis, but so far this is more or less clear. Yeah.
Yeah. Yeah.
Okay. So here you can find here you can find this paper, and you can
Okay.
find the movie and the paper. Uh,
Okay.
Sometimes you can skim some of these papers. I don't recommend you to go insane and memorize everything, because they are not exactly the topic. And sometimes, um, if you were doing a PhD, I would say, daggum, read it and learn it. For these
Okay.
days, just you can skim and say, and we can discuss about it, but not, there is no need that you memorize everything, or just,
Okay.
yeah, so don't worry.
Okay. I'm just going to take a picture of the link right there.
Oh, I will send you this. I will send you
Oh,
this.
okay.
This
Okay.
this will be, so this will be for you. I created especially for you, so
Oh, okay.
you can explore and this, and I will send you this recording.
Okay.
If we can, if I am able to record it. If not, I will record it again. But I think I will teach the classes in this way,
Okay.
because it will be easier. It will be more entertaining or I don't know. I think listen to some people talking. They said that is more interesting. But anyway, so
Okay.
look, these dots are just fascinating, because this is exactly what we see. Yeah. So it's not our imagination; they are everywhere. And the
Okay.
question is, we don't know why there are. There are peripodial cells. Uh, why those cells are moving, because you see, we don't know if there is like a current around, as they have a media.
Okay.
Are they moving? But why are they moving? Are they moving randomly? We don't know. We just know that we see the same thing.
Okay.
uh, I Later you can read about the imaginal disc. This is the leg. You remember that I told you this is like a telescope?
Yeah.
So look, imagine that this is a, sorry, this is like a, annotate. This is here is it will become the top of the distal region of the leg, and it's like a
Okay.
telescope. This is the bottom part. And the names, you don't have to learn the names of the parts of the leg. For you, this is not important.
Okay.
But the one that you know is that there is a developmental process there, that is like my PhD supervisor said, that is like telescope out, that, and that
Okay.
they have like a sock that is the peripodial membrane.
Okay.
Okay, but the information is there in case that if you read a paper and you don't understand what is happening, the information is there.
Okay.
Mhm. So I send you uh, here there is a paper that in case that you want to know more about how all the movements that happen that make the leg be elongate. And there's
Okay.
these two terms that are not very important for you, uh, that you memorize in the first day, but at the end you will say, okay, as you read papers, let me. Uh, Okay, so mhm, this eversion and elongation is equal to evagination. These are, when you read some paper, you're like, what is eversion? What is elongation? But you know that here there are good sources of information, and I don't think that there is a need for me to go in detail right now in those things. But later,
Okay.
in case that you have questions, we can go into that. But there
Okay.
are developmental processes that allow that they have been studying a lot of detail of how this happening. Yeah. And
Yeah.
what you saw in the movie is this. They go from this, uh, from like the telescope is close, and they just start elongating and elongating and elongating. Okay.
Yeah.
Let me send that to erase this. So, okay, so how they did this? It's a technique of confocal microscopy. They use here a fluorescent protein, and in this case, the fluorescent protein we use it to mark the tumors. Yeah, and
Okay.
as I said before, we use DIC, just using shades to highlight the legs and the other structures. So let's continue. And if you have this structure in green that you see here,
Yeah.
this is the periporial membrane that they
Oh,
are
okay.
breaking. Yeah, and maybe in the movies that you're going to analyze, I have pieces of the movies of the first 12 hours. Yeah, I'm going to send you ones that are after 15 hours. Yeah.
Okay.
Because in the beginning there is almost no dots. Yeah, so it's not very interesting to analyze. And at that time all the tissue is moving, so it's not very clear. However,
Okay.
then it stops moving and you can analyze more things. In case that you want to know more about the peripodial membrane that is there, blah blah. Techniques, small things about the techniques that we use. We use a fluorescent microscope that allow us to see in a lot of detail the fluorescent proteins. Yeah, so for example, here you can see you have two fluorescent proteins and you can study cell divisions. Yeah, and here
Yeah.
you have two type of fluorescent proteins. We use that, and in that way we label. We have a complicated system that is called the Markov clones. You don't need to memorize and know a lot all the details. If you look into that, you will be like, oh my god, what is all these things? What we did is we combine here. You're only describing the system, so you can have a lot of colors. Yeah,
Okay.
you remember we have 4 chromosomes in fruit fly. So in one chromosome we put one genetic construction that allow us to have GFP, another construction that allow us to make the tumors, another construction in the other one that allow us to make it at a specific time. So all the chromosomes, I wish that we can have different colors and make it more clear, but the
Yeah.
more things that we add, it becomes really complicated and it takes a lot of time. So this is why we only have one color, and I have always
Okay.
put more colors, but yeah, it's a pain.
Okay.
So, and I just want to show you a Markov clones. I wanted to tell you what is a Markov clone. First, I want to tell you what is happening when you have a genetic manipulation. When you have a wild type, all the cells are, let's say, normal. When you
Yeah.
have a mutation, like for example, if you remember all the mutations that we studied, all the cells are going to have the same are mutant. Yeah, all of them are going to miss. However, if you cannot study cancer, or you cannot study specific process if you all of or your cells are mutants. Why? Because they will die right away. If all your cells, all the cells in the beginning of the development, all the cells are proliferating excessively without
Mhm.
or without control, the fly will die very early. Yeah. So what
Okay.
we do is we create clones, and a clone is just a subset of cells. So for example, we can create a clone that have only these ones that are mutants, or we can create all that only these ones are wild type.
Okay.
Yeah, pieces of the tissue that will be different. In that way, you can study in more detail the process. Yeah, how the
Yeah.
process, how they start growing, and they survive in a better way. Yeah,
Okay.
so the Markov clone is using in a very general and very simple way is to use clones and label them with GFP. Yeah,
Okay.
and if you um, and if you want to know more, here there's more information about fluorescent proteins. What I did is I didn't have to open the pupae. I just have to put it like this in a slide in the
Yeah.
microscope and see them develop. And here they will tell you this is a normal clone, yeah, and the Markov clone will have a fluorescent protein, so we can see it, yeah.
Yeah.
And there, in this case, we use. You remember RAS that we talked about RAS and
Yeah.
Scribble. We will talk about that in cell biology.
Okay.
When you combine these two, when you have a mutation in RAS, the tissue is going to overgrow. Yeah, when you have a mutation in Scribble, the tissue is going, the cells are going to go to apoptosis. Yeah,
Okay.
and when you have the combination of those, you have an invasive tumor that is going to invade everywhere. So let me,
Okay.
here you can see RAS, you have a benign overgrowth, it's not going to invade. When you have a Scribble in your clones, the clones will die. When you have both, you will have overgrowth and invasion. Yeah,
Okay.
good. And this is the genetic background. This is how we create the tumor.
Okay. Is this possible like the ones on Scribble like they undergo apoptosis, right?
The one, if it's only a Scribble, they will
Yeah.
go apoptosis. But you remember that cancer is the accumulation of mutations. So when you have these two at the same time,
Yeah.
this is when instead of being overgrowth or cell death, this will actually produce overgrowth and invasion.
Okay. So that means the mutations in RAS like when combined to the Scribble like somehow they affect the apoptotic cycle and okay. Okay.
Uh, there is hundreds of studies about what they do.
Okay.
They start how they start studying. They did this is, believe it or not, the larvae. They start studying this in the larvae, and this is the cephalic complex. So the eye antenna, the brain, and
Okay.
the this is the ventral neural ventral neural cord. So what they start is producing tumors in the eyes, and they found when they have RAS and Scribble, they start there's invasion into other tissues. Yeah,
Okay.
so this is the normal when you have clones, but they are wild type cells. You produce here in the eyes of the flies. I just later you can see these are the eye and antenna imaginal disc. This is the eye and antenna, the brain, and the ventral neural cord. Yeah, and in the wild type they only are here. Yeah,
Yeah.
when you have RAS there is overgrowth. Yeah, when
Yeah.
you have a Scribble they is very faint, they are dying. But when you look what happened when you combine them, everything become like a huge tumor. a tumor. And this is a way that they start finding that the combination of these two genetic perturbations can produce overgrowth.
Okay. Yeah.
There is a paper about that in Nature. Fascinating, and there is hundreds and hundreds of papers. They start studying this a lot using fruit flies, and what we want
Okay.
to do is now study what happens, what are the cell dynamics. Yeah. So this is what you're going to have. We're going to have the normal, and this will be the tumors will be here. Now this is when when we start asking questions. Yeah.
Yeah.
So for example, do you see here? I don't know how much resolution you have.
Okay.
So I don't think that you will be able to see here.
Is it like gray? Are those so I can see like a few green spots here, but also I could also see some gray background.
Mm-hmm. Yeah,
Am I just imagining it or
no. And you can see later in your movie
uh-huh.
so that here,
Yes, that area, yeah.
this is a leg.
Okay.
Yeah, and here you will see a tumor.
Okay.
Yeah.
Yeah.
Here you will see another piece of a leg. Mm-hmm. And here you can find a tumor.
Okay.
Tumor. Here you find another piece of a tissue, and here you find a tumor. Yeah.
Yeah.
And here you find these pieces of tumor that are detached from everything. Yeah. When I know in the beginning you're like, Nicholas, I don't see it very clearly. You will see it, and I found that when you shared your movie,
Mm-hmm.
it was not as clear because there
Okay.
is not enough resolution to make the call. So we can talk. The resolution is not very good, but when
Okay.
you look at your movie, you will see. Yeah.
Okay.
So let me show you. Okay, since this is like a blackboard and I have to delete everything after I go to the next slide. Okay,
Okay.
so look, this when this will look like the legs, yeah, and
Yeah.
here is the other section without highlighting the legs. You will not see it in the beginning, but
Okay.
when I send you the presentation, you
Yeah.
will see it. And when you start playing with the movie, the first task that I want that you do is
Okay.
that you outline a few slides where you show me where is the lake. Okay?
Okay.
That is the first thing that you have to do. Just outline. Good?
Okay. Yeah.
And do you know how I outline things in Imaginal?
Yeah. Yeah. I've the last time you sent me the instruction for Image. Yeah, I was able to try it out. Yeah.
Good. Is that um, Imaginal has so many cool things, and if you have any doubt, you just let me know on the next call, or just send me an email. You
Okay.
can Google it, or if you, yeah. Sometimes in AVI files you cannot draw. Yeah.
Oh, okay.
So you have to switch it from AVI to TIFF, and you can draw. So you will draw. It's
Okay.
like just you do this. So I have to write that. If you draw like this, and then you go to the next slide, and
Okay.
you come back and there is nothing, it's because it's in AVI. So you have to go to TIFF.
Okay.
Save it as a TIFF, and then you can change things. Yeah. This
Oh,
is
okay.
this is in every you will see. Sometimes it's like this is so easy. What is happening? And it's because there is a new update in ImageJ that doesn't allow something. So, but you can call me and we can get expressed together
Okay.
about
Okay.
simple things. So 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 some of the questions that we can ask is, imagine you did this homework and you have here the legs.
Yeah.
The legs, yeah. And you will see, so one of the questions that we have is, do the pieces of these dots move to a specific region or not? Yeah. And we can answer that question following the trajectory, yeah, of the pieces of the tissue. So for example, here I show you, I send you a movie that there is an arrow.
Yeah.
And I manually did it. There are hundreds of ways to do it, but I just wanted to show you how, yeah, to show you an example. But this is a question that definitely you can test. Are they moving to the same region? Are they moving in every movie, in every slide? In the first slide, they move to that region and all of them move together, or what is happening? Yeah.
Yeah.
Or are they moving to different regions? They repel each other. Or they are like in the ocean, that there's like a current and they move to a specific region. Yeah.
Yeah.
And what are the implications that we can hypothesize? Maybe if they are moving to similar regions like in a current and they start accumulating a lot of cells, maybe there could be interaction, but we can see.
Okay.
The idea is that you ask some questions, and I propose the following questions. Yeah. Could you see the top of the screen? No.
Yeah, okay. I could see the slide. Yeah.
The slide. You don't see anything of the menu of
No.
Zoom. Okay, so I am the one who see it. Okay, good. So we can ask what type of cells are those ones. Yeah. We can ask. We can measure the size. What are the size? Are the size are very tiny? Are pieces of tumor? Are a single cell? What are the shapes? And you can answer those questions. What trajectories? Yeah. Do they move passively? Do they move to a specific region? If they move actively, for example, do they have a team projection that allow them to move? You know, or
Okay.
or what do we see that is happening? Are they forming? Like, you remember that they have hands? Do we, like the fingers that you saw last time in the other one? What type of things do you see? We can measure the velocity, the displacement, the direction. There is a lot of things that you can ask.
Yeah. Yeah.
Good.
Yeah.
Okay. So far, there is any question?
I think I'm good so far. Yeah.
So far is good. So yeah. Homework for next time.
Okay.
I want that you are going. I'm going to stop sharing the screen.
Yeah.
And I'm going to. Sorry. Now I have to. Were you able to open your movie in Imaginal?
Yes. Yes. Yeah.
Okay. So I'm going to do the same thing. Just give me 1 second. I just need to find. Okay. So I'm going to share again my screen.
Okay.
Ta-ta-dum. Okay, second. So just in case, I'm going to show you, and I will send you the recording in case that there is any question. Yeah. So look.
Okay.
I went here. I give you one that is with labels, and I will send you one that doesn't have any labels.
Okay.
As I said before, did you take already physics?
Sorry?
Did you already take physics?
I haven't taken physics in university, but I've taken physics in high school.
Oh no, it's okay. Is that in candidates use Imaginal to teach them something. So I wanted to know if you
Okay.
already know a lot of things, or the first time that you see Imaginal, and
Yeah, it's my first time using Imaginal.
yeah, so good. So you already know how to open it. You already open it.
Yeah.
And remember here you see that it say ADI.
Yeah.
Let me see. Sometimes there are different. I will. There are different versions of Imaginal. For
Yeah.
example, this one is called Fiji, and they are slightly different. So sometimes one things work, and sometimes in our version is slightly different. And
Yeah.
sometimes you have a Mac, I guess. Yeah.
I have Windows.
Sometimes between Mac and Microsoft, the left click is the right click, and but
Yeah.
anyway.
Yeah.
So look, I already labeled the cells. You know how to, and here you can see those pieces of tissue moving. Yeah? You can see
Yeah.
that there are some that are travel alone. There's other ones that travel in group.
Yeah.
And for example, you already you can already, for example, measure the size of each one of these. Yeah.
Yeah.
And you can also there's a lot of things that, and we can start following the trajectory. Right now, what I want that you do is the following thing. Just that I know that you are doing and that you are seeing this. I want that you pick few slides. Yeah.
Yeah.
You go here to the pencil,
Okay.
color, pick the color that you want. So the only one that don't use green and don't use black or white because or gray. Yeah, you can
Okay.
use
Yeah.
white or yeah, or let's take white. Yeah, just because if I use black, you will not see anything. You remember how to zoom,
Yeah.
and now I can go and try to just not. It doesn't have to be perfect. I just want that you show me in few slides that you know where is the lake.
Okay.
Yeah, and for
Yeah.
example, here doesn't have to be perfect. Yeah, and do it in take a screenshot and send it to me. Good.
Okay. Yeah.
And do it like in the begin, in the middle, at the end. In the last one, do it. Sometimes it go in the early development, things start becoming more difficult. So I will not start for the first slides. I will start maybe in the for the or in the middle.
Okay.
Good. And look here, you will see it will be easier where it is. And don't worry if you don't see it in my screen right now because
Yeah.
the resolution is not good, but in your movie it will be very clear. Okay.
Okay.
One thing. Let me see. Again, look. I did this. I went to the next one. I am in the slide 17. I go to the
Okay.
18. Go to the 17. You see that I cannot see anything.
Yeah.
So what happened is that as it's an AVI, I
Yeah.
have to switch it to TIFF. Okay, let's call it Dagen 1, yeah,
Yeah.
and put it in the desktop. Let me go, oh, here it is. And look, it's the same one, but look now, it's there, yeah.
Yeah.
Uh, Control Zeta, and sometimes it works to go back. In some cases, in this case, it doesn't. If for some reason you say like, oh my God, Nicholas, I have the movie and I did all these things. I ruined the experiments. Everything is wrong. Don't worry. We there is no problem. Yeah, there is no problem. If something like that happened,
Okay.
you know what we do? Look, I spent 2 hours labeling this and I screwed. So look, you take, I'm just showing you. I went here to the Control C, go to the other slide, Control V. I copy from one slide to another one, and now you don't see anything. Good?
Oh, okay. Okay.
So it's like if you have a PowerPoint doc or a PowerPoint document, and you can copy one slide from the other one. It's one picture from the other one. So if you find any problem or anything, or just tell me and we fix it the next time.
Okay.
Okay. In the beginning, if you have any question, you can give me a call, and 10 minutes Nicholas, why this is not working? I say, oh, I forgot to explain something, and it will take you a minute. Okay.
Okay.
Generally, our meetings will be much shorter, but
it's fine. It's fine. Yeah.
But yeah, it was a pleasure talking to you, and have a good day. And I'm very excited with this project, and hopefully everything goes well.
Okay. Okay. Thank you, Nicholas.
Okay. Have a good day.
Okay.
See you.
Bye.